All right, so let's say you just started volunteering in a lab and you've been told that your project revolves around studying a certain gene. So where do you start? How do you even start? So just like with any intellectual endeavor, we have to start by looking back at the work of what people have done previously, right? To find out what we know and what we don't know. So this sort of information, if we're thinking about a gene, this sort of information might include the sequence information of the gene. So this might include the DNA or the genomic sequence of a gene, or maybe the mRNA sequence of a gene, or even the protein sequence of a gene if that's applicable. Other information that we might find useful would be the known biological function of a gene, maybe even what the protein binds to, perhaps what it looks like, or if the protein itself catalyzes some sort of chemical reaction. And to make a long story short, there are so many other things that might be known about a gene that might potentially be of use to us. So this might include where these genes are expressed in an organism, their evolutionary relationship with one another, and so forth. So where exactly do we find this information? Where do we even start? So when scientists discover something new, it usually will go into a reference database that's available to the public. And really knowing about and being able to use these sorts of online resources is really essential to do any sort of research in molecular biology. You may have heard of some of these resources in the past. The best known collection of databases in the biological sciences is maintained by the National Center for Biotechnology Information, or NCBI, which is a division of the U.S. National Institutes of Health. Although it's called the NCBI, scientists often just refer to it informally, and this is also incorrect, but they refer to it informally as PubMed, which is just the publication search engine, which is maintained by NCBI. Now, for the purposes of what we're going to be doing throughout term, we'll be using a database maintained by the U.S. Department of Energy's Joint Genome Institute. This database can be accessed through an online portal called Phytozome. So let's go through the use of this database and figure out how to obtain the sequence for the open reading frame, or the protein coding region for your gene of interest. So when you access the Phytozome portal, and you can see the URL up in the address bar here, you'll see a quick search box here where you can query the database for your gene of interest. So you'll want to start to define the species that you want to search for here, and indicate your gene of interest here. So in our case, we'll search for something in the species glycine max, which is more commonly known as soybean. So we'll put that in here. And you can see that it automatically populates. And let's search for, I don't know, let's search for a gene that you guys have already heard about before, lactase. So lactase is a gene that codes for a protein that's responsible for the degradation of lactose, the sugar. So we'll put that in here. And we'll hit go. And so this brings up a bunch of search results, as you can see, which corresponds to our search parameters. So one of the things that you may be wondering about here is, why are we seeing so many hits come up? Why are we seeing beta-galactosidase come up as a search hit when we looked up lactase? Well, genes will often have multiple names, and lactase is also known as beta-galactosidase. So that's what we're seeing here. You'll also sometimes pull genes that are related in terms of function, which is the reason why we're seeing some of these other search hits as well. Note that if we had searched using a specific gene identifier, we should only get back one search hit. Now here you can see a brief description of your search hits. And to the left in these little green boxes are links that provide access to various forms of information for your gene. So what we're going to do is we're going to navigate to the genomic information for one of these searches by clicking on the green G naturally. And this will bring up the information for that gene. So I'm just going to pick, let's pick this guy here, beta-galactosidase 5. And when we pull up a specific gene, there's a lot of information that you'll see here. Some of this will include the unique identifier for the gene and other names that this gene goes by. So you can see this here under the gene name. The transcript name is shown here, and there's some aliases that this gene goes by as well. And by checking out other parts of this page, you can actually find other interesting information, like what others think this gene does in a cell. And so that's shown here on this first page, and you can get to it by clicking on functional annotation here. But once you open this up, it kind of shows you what others have found that this gene is responsible for. You can also find out where exactly this gene is expressed in an organism. So if you click on the expression tab here, you can see that others have looked at specific tissues and made the determination as to whether or not this particular gene is made in those tissues, which is interesting information if you're interested in studying this gene in depth. And also, you can find information on the alleles of this gene that exist in nature. And if you go to the variation tab, you can pull up the frequency of specific alleles for this gene. So the amount of detail that we can get for a particular gene is actually pretty incredible. And there are many other things that you can see. So I encourage you to look around. You can kind of click around. Don't worry, you're not going to break anything. So don't worry. But in our case, we're interested in finding the sequences for this particular gene that we're looking at. So if you click on the sequences tab up here, you'll see different types of sequences that you can access. So there's the genomic DNA sequence, the mRNA or the transcript sequence, and even the protein coding sequence, which is abbreviated as CDS. So if you click on that, the CDS sequence, you'll see the protein coding sequence of the gene pop up. And you can see that it starts with the start codon, ATG, and it ends with the stop codon, TGA. So this sequence here is really the starting point for any lab cloning project. So if you decide to join a research lab on molecular biology sometime in your professional career, you'll likely be asked to do something like this. Find the sequence of a gene because you need it in order to study it, right? So there you have it. This is how we can use a genomic reference database to find the protein coding sequence for your particular gene of interest. As you can tell, there are many other pieces of information here that we can't cover in the limited time that we have together. But again, I encourage you to take some time and poke around and explore here and learn more about the gene that you wish to study. And you can find the user guide to Phytozome at the link below. And as always, feel free to reach out if you have any further questions.